Welcome to Nikolay's genetics lessons, and today I'm going to explain how to feed computers with information which computers understand about RNA folding. So today I'm going to show you some basics of bioinformatics. Today's example is very simple, but in my following videos I'm going to use more complex RNA structures. Every single RNA molecule starts with 5' end and ends with 3' end. So our first step is going to be to transform this two-dimensional structure of RNA into linear form. So we have at the 5' end adenine here. So let's also add it here, adenine. Next goes cytosine, then uracil, then guanine, and adenine, and uracil, cytosine, guanine, uracil, adenine, guanine, uracil, and cytosine, adenine, and cytosine. As you see, first nucleotide adenine also base pairs with uracil, which is, let's count, 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11th, and 12th nucleotide in this chain. So let's bridge them on this picture. Next we have second nucleotide cytosine, which base pairs with guanine. So also let's connect them. Next goes uracil and adenine. Again, let's connect them. So, and the last couple is going to be guanine and uracil. Also, let's connect them on our graph here. Of course, nowadays computers can work with any pictures and convert them into anything we want, but at the beginning of the bioinformatics, scientists have to convert this graph into something that computers would understand. This is how dot bracket notation formed. So take a look. Again, we have 5' end here, just like in this linear form here. But next, take a look. Let me highlight it here with green color. So, do you see? It looks like a bracket, opening bracket. So we put opening bracket here as well. And this also looks like brackets. So also let's put opening brackets 1, 2, and 3. Next, nucleotides A, U, C, and G. As you can see, A, U, C, and G here doesn't have any other nucleotides to base pair with them. So we represent them as dots. So 1, 2, 3, and 4. 4 dots. Then we have 1, 2, 3, and 4 closing parentheses. Let's add them here as well. 1, 2, 3, and 4. Then goes these 3 nucleotides, which also doesn't have any pair, so we just represent them as dots. 1, 2, and 3. And the last is going to be to designate 3' end. So as you see, we just converted this two-dimensional RNA structure into format which computers understand. Base pairs can be coded by matching opening and closing parentheses, and unpaired nucleotides can be represented by dots. With this format, computers can work and convert them into the two-dimensional and then following by three-dimensional structure of RNA. And this is all for today. Subscribe and see you in the next video. Goodbye.